Hello, everyone. I have the honor to share this panel with a lot of esteemed scientists and longevity advocates. If you allow me to briefly introduce myself, I'm Kris Verburgh. I'm a medical doctor. I'm a consultant, also a researcher into aging, and I also am a partner at a fund that invests mainly in longevity companies. The other panel members, you have already been introduced to them because they have given talks. Besides one person, Sarah Constantin of Daphnia Labs. So if you could briefly introduce yourself and explain what you're doing, in fact.
All right, sure. Okay, this one. All right. Hi, I'm Sarah Constantin, and I recently founded a biotech startup called Daphnia Labs, which is looking for drugs that delay or prevent the diseases of aging in a small model organism. I'm actually a machine learning person by training, and this can be a machine learning problem if you can have a visual phenotype by putting a camera on a small model organism and observing its behavior. How do they stay moving over the course of their lifespan or not? So we're trying to discover drugs that hopefully make people live healthier lives. And in parallel, about a year ago, I started a nonprofit that's funding research into lifespan studies in mice that's trying to replicate the large effect size results that have already been in the literature but maybe haven't been independently confirmed by someone other than the initial scientists that discovered it. So this is a grant-giving organization because there's a gap in funding for lifespan studies in mammals, and we've been collaborating with Ichor on that quite a bit as well. So thank you.
Okay, great. Just wondering, the Daphnia Labs, you study like these tiny little model organisms. They're like sea fleas or
right.
water fleas, zooplankton.
Yes.
How big are they? Like 1 millimeter? Or
1 to 5 millimeters when they're full grown.
All right, great. And why do you think they are a better model? Because normally they use C. elegans for very early studies. So why do you think perhaps your approach would be better than using C. elegans for these kind of studies?
Well, the major advantage is that they are very sensitive to drugs. C. elegans is traditionally a genetic model organism. That's the beginning of this whole biogerontology field with Cynthia Kenyon and Tom Johnson, that there are genetic modifications that make them live longer. But they're not necessarily a pharmacology model organism or a toxicology model organism. It's very hard to poison a worm. It's very easy to poison a Daphnia. You're learning something immediately about, this is a standard organism for testing what's safe to put in the water supply. They're very sensitive to drugs in their aquatic environment, and so that can tell us maybe more than we expect to be able to get from C. elegans.
Okay, great. Thank you. So I have a question for each panel member, and the first question would be, in fact, what do you think is the most promising technology or approach to address aging? So in your opinion, there are many approaches. We have seen many today and yesterday, but if you had to choose one, which approach do you think is most interesting or promising to address aging, and why? So I will perhaps start with you, and then we go one by one.
So I'm a SENS advocate, as I think most of you know. So the question is given what we know about SENS to date, is it even sensible to try to answer that question? And I think, as we all know, Aubrey himself was rather skeptical that, let's say, clearance of senescent cells was going to do great, meaningful things before it was actually done. And actually, yes, it does do great, meaningful things. And we don't know a lot about the interactions between these items either. If we want to look at what would be influential coming up in the near future, and I'm going to do the political thing and not quite answer your question, but say something instead. There's this question of what causes skin aging in 20s and 30s, because it's clearly not senescent cells. Yet there is something going on. So is it crosslinks? Because if it's crosslinks, then the next 10 to 15 years are going to be really, really interesting for the quote anti-aging unquote industry, because suddenly they will have something that works for all those young people who are rather angsty about what their skin is doing as they head towards age 30. So that, I think, will be a big deal. But as to the rest of it, you're really not going to know until you do these things as to what is going to be most influential. One can argue for each one of these SENS strands that, yes, this will be absolutely amazing it will do great things, or one could make reasonable arguments to the contrary, and then while you're doing that, I will stand there and say, yes, but we got senescent cells completely wrong. So I think you're full of it until such time as we demonstrate this thing.
All right, and just to catch quickly on that, you think senescent cells don't play a role in skin aging. Why, in fact?
Oh, they do. I mean, obviously we have OneSkin who are doing exactly clearing senescent cells to treat skin aging. But if you look at senescent cell burden, what we know of it, what we think is going on, the logical conclusion is that the burden of senescent cells scales in the same way as cancer risk does, and for the same reason, that your immune system is declining in its surveillance. So I do not think, based on what we've seen from the self-experimenter community, that senescent cells are at all influential in aging prior to, let's say, 50s, 60s, depending on how healthy you are, because that's the age at which you start to get significant bumps in your cancer risk. And we can argue that that's because of immune decline. So this is all, of course, speculation and hypothesis until such time as we have really good assays for human senescent cell levels, but it's eminently reasonable as a point of view, I think, being the person who's saying it right now.
All right, thank you.
Okay, so I guess I'm going to parrot Reason's opening statement of diplomacy in saying that, it's obviously very difficult to try to say that you have the master plan of exactly what should be performed when and how everything's going to come out. But I guess what I would like to say that's not a complete dodge, somewhat related, is a tactic that I would like to see more increasingly used is, historically, research and, clinical therapy development happens in silos. And I think that, aging is a multi-factored problem, and I think our system needs to be more amenable to looking at combination therapies and combination testing, and, to be able to maybe involve new ways of doing clinical trials, like that could have a crowdsourced element, because you can imagine that there could be some combinations of 3 or 4 low-hanging fruits of things that could be maybe already safe, generally recognized as safe. And if you can do a crowdsourced clinical trial in a way, you might be able to get a high level of statistical power just from the cohort size, even if you're losing statistical power in other ways.
Okay, and what kind of low-hanging fruits are you thinking of?
Like we've talked about things like, fisetin and strawberries as a, potential senolytic, and there are other therapies that could be seen to maybe partially remediate dementias in mice, things that sound too simple and too good to be true, like lights of a flickering at a certain frequency and things like that. And you can imagine how in the age of cell phones, where a lot of physiological biomarker data can be collected by something you have in your pocket, you could probably structure some kind of trials in very interesting ways where even just the way in which you are collecting the data ensures that the protocol is being conformed to. And I think there's a new wave of that could enter the mix that could be bring a lot to bear on aging.
Okay, fascinating. And there are a lot of companies also capitalizing on that. You have like companies that look at the movement of your eye from your smartphone to see whether you are at an increased risk of getting Alzheimer's disease and so on. Even companies that are being founded that will track your how you swipe and then see whether you're at an increased risk of neurodegeneration.
You're even seeing some interesting overlaps of fields that you wouldn't expect. Like I think there was an iPhone video game that is a game, but it also tracks how well someone remembers pathing, and that has potential relevance to Alzheimer's diagnostics and things like that. So I think, there's a brave new world of multidisciplinary combinations that could be interesting here.
All right, great. So Sarah, what do you think would be an interesting technology or approach to tackle aging?
So there's a lot of good approaches out there, and just going meta, I think that you don't. It's the track record of trying to pick winners a priori is very bad. You think you have a target, you think you have a mechanism of aging, you think you have an intervention that will do something about it. More than 99% of the time, you don't. And that has nothing to do with the quality of the science. That's just the difference between hypothesis and experiment. That's sort of the partisan take the position that I take is that that's why the way you screen is very important. And that's why I believe in phenotypic screening. So, in other words, what is your outcome metric is very important, how to select it, and the hardest measure to gain, the most reliable indicator of aging is aging. Some kind of measure of function, motor or cognitive function or lifespan or some kind of the thing you're looking at. So sometimes you may be able to find a very good proxy. We have some very interesting proxy measures of the kind that with the expression signatures that have been talked about today. Maybe you can get the correlational proxies just right. That might also be good. But ultimately, what are you testing is a harder and more interesting problem than which particular pathway, I think, because you're not going to know ahead of time.
I have no idea. That's why I have a portfolio of companies instead of just one.
Great. All right. Wonderful. All right.
No, I'll take a crack at it. So I don't think it's, certainly as the other speakers have indicated, it's obvious at all what could be a major driver of aging. And as Reason pointed out, there's a lot of arguments for and against different targets. So a couple of things that we look for, at least at the level of program selection. I think that target validation is something that is highly underrated in the aging community. I'm really excited and enthusiastic about the talks I've seen at this conference because I feel like there's a significant maturation, not just in going after the right molecular targets, but defining right as targets that have been validated in all kinds of different ways. So I do feel that the efforts that are being put forth are perhaps a lot more reasonable, a lot more focused, and going to generate more tangible and actionable endpoints than what I kind of saw when I first entered the space, and that's highly encouraging to me. The entire point of Ichor, right, is to be this ubiquitous thing that can be deployed readily on different projects as our understanding of the space is further expanded, ideally in a way that larger governments or larger pharmaceutical companies lack because they're much bigger entities. So that's my long-winded way of saying that I don't know what the best targets are, I don't know what the best things to bet on are, but I do think that if you couple an aging pathway with conservative indications, you can meet your fiscal responsibilities to investors while at the same time adding a lot of clarity to which targets are best to go after. Is that okay, Mike?
Is that okay? I have the advantage of having thought about this the most. Is this working? Yeah? You can hear me, right? Yes. I can't hear me. There you go. Anyway, so I've had the opportunity to think about this the most. So I think of it in two ways. One, what's the most promising approach for people, for humanity right now? And some of those of you who know me, I don't have to give this sort of disclaimer to, but I'll just say it anyway. I hate death, okay? So I'm sort of an immortalist. I have been my whole, I've been a closet one. In the old days in aging research, you could never talk about lifespan extension. You'd be kicked out of the lab, I mean, really, seriously. And, but having said that, we have a social responsibility. And in the United States and in a lot of countries around the world, thinking about implementing a therapeutic that would extend human lifespan 20%, 30%, or even doubling or whatever, it's too late. The boomers are already old. We need to apply these technologies to degenerative conditions that alleviate human suffering that people are experiencing right now. And so I would argue the most promising clinical approach are regenerative therapies, something that's going to help the patient not get more blind from macular degeneration or alleviate the pain of arthritis and so on. On a scientific level, completely different side of the coin. Again, being in the field a long time, I feel like when I was a boy, remember that? We walked through the snow and went to school. So I called a guy named Sam Goldstein, who was a pioneer in aging research when I entered the field, 1981, I think. And he could tell I was interested in aging, and very committed gerontologist, wonderful man, and gone now, And he He says, so what are your thoughts on aging? What do you think is the most promising approach on aging? He asked me the same thing. Long ago was that? Long time ago. And going on 40 years ago. And I said, okay, so I got an idea. So the fall, leaves fall off the tree, right? So there's cells that I thought I was real smart, you know, that hold the leaf to the tree, and those die, and maybe we could learn something about human aging. And he interrupted me. He says, okay, look, Mike. He says, you're going to spend your whole career, it sounds like, working on aging. Shouldn't you probably focus on humans, right? What if you spend your whole career studying leaves and it has nothing to do with animal or human biology? And I thought, oh, shoot. You know, immediately changed my thinking. And so I would argue that the most promising scientific approach is to study something like progeria and Werner syndrome that are showing the manifestation, many of the manifestations of aging way too early, and they point toward, you know, the status of the chromatin, DNA integrity, chromosomal integrity. And I think that's a major clue that nature's handed us on a golden or silver plate, golden plate, whatever, both. And so I would scientifically, studying chromatin integrity, I think, is really where the action is.
Okay, interesting. Of course, you have these aging diseases, accelerated aging diseases like progeria, Huntington-Gilford's disease, and so on. But they see it's a defect of the nucleus, and it causes a lot of things that resemble aging. These children, they live for 13 years and so on. But it doesn't entirely encapsulate aging. Most progeria diseases, for example, progeria patients, they don't get Alzheimer's disease, they don't get immunosenescence, they don't get cataract. So there is some discussion in the scientific world, like is Huntington-Gilford a good model for aging, or not? So.
Yeah, but the lamin mutation in here is not expressed in neurons, so, and a lot of the CNS anyway, and so it couldn't possibly, the mutation couldn't possibly, the gene's not expressed there, so you wouldn't expect necessarily aging in the brain.
All right, great. And because you are you mentioned regenerative medicine, because it's like one of the concrete answers of this panel. So as a medical doctor, a lot of medical doctors, they have some reservations. I strongly believe also in the approach of regenerative medicine. But a lot of my colleagues, they say, well, regenerative medicine, stem cells and so on, they have a lot of shortcomings. If you inject the stem cells into the body or the bloodstream, because of the shock of the injection, most stem cells already die. Because they circulate in the bloodstream, for example, most of them are destroyed in the lungs. The lungs that survive are destroyed by the immune system, and the graft also very difficult. So how would you try to solve these issues? Because that's especially the grafting.
You didn't give me any time to think about that one. So, all right. So, well, right. So no one, I guess I can't say no one, but the concept of the stem cell thing with pluripotent stem cells, as I alluded to a bit in my talk, was that these cells are still in the germline, basically. So they have telomerase, and when they start to differentiate, the cells are incredibly young, right? I mean, younger than normally we have access to in human biology. We never intended to inject embryonic stem cells into a patient because they would just make a mass of different tissues. It wouldn't be appropriate tissues. And we certainly didn't manage injecting them into the blood. So there's a lot of confusion. So what a lot of people are doing to make money, and this is really snake oil stuff, but you know, people in aging research, I mean, this field has been crowded with snake oil and will get worse. So as the work that everyone in the room is doing becomes more publicly known, the people are going to come out of the woodwork saying, you hear about all this advances in aging, we got it now, right? So what happened in stem cell research is they said there are these bad people, I won't name names, of course, after calling them bad, is they said, okay, look, FDA doesn't regulate autologous therapy where the cells are minimally manipulated. So we can take some fat from your abdomen, liposuction. If all we do is centrifuge it, we really haven't manipulated the cells. You can get a nice pellet of cells. What the heck? Let's call them stem cells. Why not? Yeah, that's it. They're stem cells. Come get them now, and they inject them in the blood, And you're right, they get caught up in the capillaries of the lung, and they rake in the money. But that's not, that's pseudoscience.
All right, great, thank you. So we all know, everyone in the room knows that if we can address aging, we will address many aging-related diseases simultaneously, ranging from heart disease, Alzheimer's disease, macular degeneration, osteoporosis, and so on. So a general question, when we find therapies to really go to the root cause of aging and at the same time address all these aging-related diseases, of course, there are so many aging-related diseases, but which ones do you think are the first ones that will be, let's say, cured or substantially improved or ameliorated regarding aging-related diseases? So which, in the near future, which aging-related diseases do you think this approach of tackling aging itself will get bear the first fruit of this kind of approach?
Well, okay, so let's start from first principles here, which is that the only fundamentally useful way to treat aging is to repair underlying damage that causes aging. Everything else is just messing around. So, I don't think there is an age-related disease that is caused by just one form of damage. I'm willing to believe that anyway. I mean, we can argue about arthritis and senescent cells, but I'm sure there are other mechanisms in there. Crosslinks seem important in cartilage, for example. So I think from that point of view, one can say, if you just fix one thing, you're going to cure nothing by the fundamental definition of cure. You will maybe push back and slow down considerably and or reverse those conditions that are very heavily dependent on the one thing you just fixed. So we're seeing this shakeout now for senolytics in the IND discovery process as people choose what to work on. And some of that is because, obviously, arthritis seems very, very dependent on senescent cells in the sense that you can cause it with just senescent cells, even though I'm willing to believe that it's not the only contributing factor in the wild-type condition. So given that, what do we think is going to be first? Well, evidently, it has to be something that is going to be largely cured via removing senescent cells, because we don't really have anything else from the SENS portfolio that's coming in anytime in the next 5 years or so. Unity will be through their first trials by the time that gets going. Not that I think greatly of Unity's current approach in comparison to some of the others, but that might just be talking out my book. So the argument here, I think, has to be arthritis, inflammatory bowel disease, or something else where it's very clearly that the dominant mechanism is inflammation caused by senescent cells, and in which there doesn't seem to be a great deal of influence by some other process. If somebody else on the panel can come up with something that fits that description that is not either inflammatory bowel disease or arthritis, please go for it.
I'm going to not do what you just said by agreeing with you. I pretty much agree with Reason on this, that senolytics are obviously one of the hot topics now, and for good reason, and arthritis will probably be one of the first ones on the list that will be taken off the board. So given that, I want to pivot this question a little bit to what, in an ideal world, what should be, or what would I like to see as one of the first areas attacked on aging. And for me, for those of you who are at the X Prize event that I had mentioned in my opening talk, I'll parrot what I said there, which is neurodegeneration, Alzheimer's disease, dementias. Because I think that there needs to be a more reasoned and thorough discussion about what the exact impacts of diseases are, especially when you can parcel them out into a specific category like neurodegeneration. Because, for example, let's compare it to heart attacks, for example. If you get a heart attack, you might die in an instant, and that's obviously very terrible, right? But if you get protracted Alzheimer's disease, your family is basically destroyed for 10 years. And that's not just on the person who's suffering and the caregivers, but also massive socio-economic drains on society. And also a very important aspect of this, I think, to bring up is that I actually think Alzheimer's plays an outsized role in what Aubrey calls the pro-aging trance, a sense of learned helplessness with how we can deal with aging, and that it's possible. Because I've had this conversation many times where I might be telling someone about our work as a field, and you'll get a reaction like, come on, you're going to address aging? You can't even cure Alzheimer's disease. And I think that actually has a very big impact, and if collectively as a field we can address that in any meaningful way, it will send massive shockwaves throughout society.
All right. So a tactic that I've seen a lot that I think is interesting and promising is you're trying to target aging as a whole, but your first indication is a rare disease. Basically, there's a lot of advantages to rare diseases as a first thing to go after, because sometimes you know the particular gene causing them. And also, the communities around a rare disease are extraordinarily active and committed. You have some of the most impressive citizen science I've ever seen has been from families of children born with rare diseases. So I've seen examples of researchers who are going after mitochondrial disorders as an avenue towards doing something about mitochondrial decline with aging. So it's one of many possible approaches, but I think it's interesting.
I go back to my original comments on target validation. I think the point of orphan diseases is well stated. When we're looking at programs, one of the things that we're most interested in is there an accelerated onset version, a genetic version of this disease that gives us confidence that the target that we're going after is causative in the first place? Because it's very easy to find associations or correlations that are not causative and spend a lot of resources removing a biomarker that isn't actually involved in the pathophysiology of the disease or so on. I actually, depending on how you would classify it, Reason, I like infectious disease for aging studies, and I know you're a little skeptical about Restore Bio's rollout with the rolling eyes and so on. But the idea of treating chronic age-related diseases, it's very challenging to do because your clinical trial duration is so long. It takes a long time for disease progression to occur. So I like about the orphan disease approach, or also an infectious disease approach, if you're fixing immunosenescence or something like that, is your feedback loop to get actionable information is a lot faster. You're de-risking the opportunities both for your investors, but then you're also making sure as a scientific group that you're following the data to make the best decisions possible. For virtually all of our programs, we're trying to include either infectious agent or orphan disease correlates. I spoke earlier about age-related macular degeneration, but certainly we're also intending to approach Stargardt's macular degeneration as well, which is a juvenile onset form of the disease, precisely for the reasons that Sarah described. I think most importantly, we need to choose indications as we develop drugs for aging that can get us to very clear no's or very clear yeses in terms of target validation. Back to macular degeneration, we don't actually know if lipofuscin in the retina causes the disease. We've got a lot of reasons to think it might, but no one's actually removed it before. And what I'm really bullish on with that as an example program is if we remove it and it fails, it's a definitive failure that is not a target worth going after. And even if we fail gloriously, we'll still have de-risked using non-human enzymes in people to augment lysosomal function in the same way that even though we're not seeing fantastic clinical results with removing amyloid plaque with antibodies and so forth, we are de-risking a lot of different approaches, and we're starting to get some definitive data about that as a viable clinical target.
All right, interesting. And perhaps if I can catch on that, because I hear infectious diseases coming up a lot of times, treating infection, reducing inflammation, in fact. So there are a lot of approaches to do that. We have seen studies where they create transgenic mice that are modified to have less inflammation going on in their body. They live longer. You kind of modify the microbiome. You even have a recent study where people exfoliate or moisturize their skin, and they have less inflammation going on in their body, because the skin is also important as a barrier, and when you get older the skin gets thinner and you have more inflammation in fact going on because of that process. So there are a lot of ways to address inflammation in the body, but of course inflammation is more a secondary effect of underlying root causes of aging. So in that sense I think it's if you really address the root causes of aging, then automatically you will also improve inflammation. But nonetheless it's good, and we even see in people, in patients, that taking anti-inflammatories, they have less risk of Alzheimer's disease. So there are many ways to reduce inflammation. Of course, some people perhaps take baby aspirin, a small dose of aspirin. There was to also reduce inflammation, perhaps to live longer, despite there was a big study in the New England Journal of Medicine showing that didn't really extend lifespan. But yeah, I believe that inflammation is very interesting, but it's more a secondary effect of all the underlying aging mechanisms. Great. And
Yeah, so I would think macular degeneration may be one, because I think the retinal pigment epithelial cell layer is the fault. Years ago, we measured telomere length in the RPE layer of cells and compared it to the neural retina, which presumably does not replicate with time. And we saw indeed an age-dependent decrease in telomere length in RPE. We never published that data, but just because we were busy. And so there's lots of reasons to think that the senescence of RPE is causative. And with pluripotent stem cells now, there are numerous groups putting very young RPE cells back into the retina. And some of the data being shown in meetings is very encouraging, that the cells can reform a monolayer and function. You can see the cells using modern imaging technologies. You actually see the individual cells, and it looks very promising. So I think there's end in sight for blindness from age-related macular degeneration. What I'd like to see, which is not being done, is hematopoiesis. Years ago, we did an experiment we called the old cow experiment. We took a geriatric cow and cloned it, therapeutic cloning, and made a little fetal clone, and took from the fetal liver hematopoietic stem cells and dropped those genetically identical but rejuvenated cells into the blood of the cow without any myeloablation, just exogenous added cells, and we got the bone marrow was really fatty and old, but when we added these cells, they robustly engrafted, and it was shocking. That was a negative control we didn't think that they would take. And it suggested to us that, if someone were to develop these regenerative, IPS kind of program for hematopoiesis, not only in age-related immune dysfunction, but even things like HIV. We now know you could cure HIV by engineering in AIDS resistance and then reconstituting an immune system in AIDS patients. So for numerous reasons, I think that would be a wonderful thing that, to my knowledge, is not being done correctly.
There is actually someone in our community working on that.
Wanting to do it, yeah.
Jeremy Esler, I think, ship of Theseus, works with Icaria.
But it's like him and very small effort, right?
all right, great. Thank you. So let's discuss some hurdles to implement all these fascinating technologies we discussed during this meeting into clinical practice and to make them available in hospitals and in the medical practice of physicians everywhere in the world. What do you envision as some important hurdles to achieve that goal, achieving these longevity therapies that really address the root cause of so many aging-related diseases in the hospital, in the medical practice, perhaps at home, and preferably worldwide? So how can we achieve this? Are there some ideas or suggestions or hurdles we need to overcome?
Well, let's think about money as the cost of therapy as the important factor here. So what does aspirin cost? Not really a great deal. How many people use aspirin? It's certainly not all of them. So as and when there is a rejuvenation therapy that costs pennies per dose, which maybe 15 years from now, once somebody gets to seriously processing dasatinib down to how am I mass manufacturing this thing for every human being, Will everybody use it? No. There's a certain limit to what you can plausibly achieve. At the other end of the scale, what about gene therapies that will never ever crush down below a few tens of thousands of dollars? And cost much more than that now, but even at scale, they will never get much cheaper than that, most likely. How many people will they be used by? How many people right now use therapies even intermittently once every decade or whatever that cost tens of thousands of dollars? And the answer is not very many people at all. So I think there has to be some very, very major change in how this all works, because, I mean, some of those tens of thousands of dollar therapies, they do actually really help, and they will extend people's quality of life, and they will extend life in general, even if it's something where you're looking at, okay, this person is horribly sick, and we're going to do something about that. You know, does everybody use them? Who should? No. And that's an interesting question. Do we actually need to do that? Are we ethically obligated to do that, or is it merely a matter of offering the choice?
Well, I am going to agree that funding is obviously one of the major bottlenecks, which I'm sure many people will share that sentiment. But I think a part of this that is often overlooked is the co-present issue of public sentiment and how that is very related to overall funding in a field. And I'll go back to an example I think is very illustrative of this, the work of the early cancer advocates in the 1940s and 50s and so on, where, this was news to me when I first read, it was Emperor of All Maladies by Siddhartha Mukherjee. I grew up in the 80s, and I thought, yeah, America fighting cancer was just something that we always did. And, it was very new. It was shocking to me to learn that in the 40s, if someone had cancer, you just sort of put that person in the corner and no one talked about it. And it really transformed from the work of very few people, notably Mary Lasker and Sidney Farber. And what was interesting is that they seemed to follow, whether intentionally or not, what looks to be a very clear roadmap. They identified what was the most credible work at the time, early chemotherapy, as sort of a credible call to action. And then they essentially did what you can only call slick PR maneuvers around that, like the Jimmy Fund, if you remember when they were doing telethons, and again, I'm not sure how intentional this was, but they sort of leapfrogged a cognitive bias that we have to deal with in aging as well of, it's very far away, it's hard to identify the victim, and they cut right through that with the Jimmy Fund, where, all right, forget about this amorphous thing called cancer, let's try to save this boy, Jimmy. And they had all these people like calling and donating, and they built up all, maybe they raised a few $1 million, just like with crowdfunding now. It's not going to be the amount that the government can allocate, but it's enough to get attention, to get some actual, progress happening, and then catalyzing that into a grassroots movement that unlocks serious government funding. So for them, it was the 1971 Cancer Act, where they basically were taking out like full-page spreads in the New York Times and the Washington Post that basically were saying like, hey, Nixon, what the hell are you doing? And then eventually that led to the 1971 Cancer Act. So I think there's a role for that here, where if we can do what we do very publicly, that's why I'm always harping on being communicators, not just scientists, advocates, and investors. Let the world know that this is important. Let the world know that this is feasible. And maybe I'm a little bit more optimistic than you on this, where I feel like if there is, 10 years down the road, some therapy that really has significant results, even if it does a silly thing like turn gray hair brown again. I think second you see your neighbors doing that, you're like, I want that too, what's going on? So I think that is the aspect of this that I think can really help unlock the main bottleneck of funding through public sentiment and awareness.
So I think that it's true that cost is one of the major bottlenecks. So when you're thinking about, can something be a aging therapy that helps everybody, billions of people, live longer, healthier lives? You have two prerequisites, and this sort of is a very interesting bar when you look at all the things that are putative therapies out there. Do they meet these things? First of all, of course, it has to work, but it has to be affordable, and it has to be safe for a relatively healthy person. So chemotherapy is a really great example of how they had an impressive hurdle to overcome, because the early chemo drugs, they almost killed you. Chemo's not fun now, but at the very beginning it was deadly. And the only reason that they managed to get through it was through incredible courage, and also because the patients would definitely immediately die if they weren't given chemotherapy, which might also kill them. So if you're looking at a putative anti-aging drug, and you're talking about giving it as a preventative or as reversing age-related damage, but in a relatively healthy person, not someone who's terminally ill, then it has to be safe. It has to meet a pretty serious safety standard. If you want everybody to take it like aspirin, it has to be as safe as aspirin. And that means we also have to test it for safety in healthy people at some point. And that's a challenge, which I know I talked about very special cases of rare diseases and so on, but sooner or later you have to see, do you have something that you can give to somebody who isn't deathly ill yet?
Yeah, if I can catch on that. As a medical doctor, I'm very concerned about safety, and you have people like taking metformin, but we know in some studies that it could increase the risk of Alzheimer's disease. We have seen also the talks about taking these HIV drugs that could reduce the retrotransposons jumping around in your DNA. But we know that also the people who take these HIV drugs, they don't just inhibit the retrotransposons jumping around, but they have also a lot of other effects on the body, like they inhibit polymerase C, which decreases mitochondrial multiplication. So a lot of these patients taking these drugs, they get a lot of side effects, like problems with the neurons and with the nerves and so on, and they have higher increased risk of diabetes and so on. So I see a lot of aging scientists very enthusiastically talking about one specific molecule and only focusing on one aspect of aging, but yeah, the side effects and other things, because contrary to Jimmy, who will die of cancer, aging is something like something a lot of people consider as a normal process, and we really have to make sure it's very safe, because we have to apply it for a long time, and I think sometimes we jump too fast to specific conclusions. So, and I think a lot of medical doctors are very worried about that issue a lot. Do not harm is the most important thing we have, and we have to be very sure that these aging drugs don't do that. So thank you. So perhaps...
Could you restate the question?
Yeah, the question was, well, what do you think are some hurdles or problems we need to overcome to implement these therapies in the general practice of offices of general practitioners or doctors or hospitals everywhere in the world?
I think in a very boring way, a lot of that does boil down to just manufacturing, which is why, I mean, we know that there's a lot of stem cell therapies that work really well, potentially, or at least they have promise, but how do you overcome the manufacturing hurdles? Gene therapies, we've got a lot of very exciting preliminary data in various animal models, but how do you manage to manufacture that on scale, which is why for a lot of programs I'm kind of bullish on small molecules. In terms of advancing anti-aging therapies in general, though, one of the things that I've kind of seen quite a bit is there seems to be a bit of a chasm between people that know how to do drug development and academic researchers that are discovering things and developing things for the first time. I think probably my favorite talk of all of this conference so far was, I think, Novo Therapeutics. I hadn't seen them talk before, and that was a beautiful presentation of an advanced stage preclinical program with all the check boxes marked, and that's something that I don't see enough of in this space. A lot of people, half the events you go to, and we're talking about offshore clinics and why that may or may not be a good idea, emphasis on the may not be a good idea. And so there is also a certain element of sophistication, I think, that needs to exist in order for the field to move forward, people that have taken drugs into the clinic, people that understand how to balance the risks to patients versus the need to bring in new medications, and people that understand how to go through a regulatory process in an expedited but responsible way to address some of the things that you guys are talking about. So the two things that I'm most focused on at our company are manufacturing considerations, because it's a boring topic that no one I can't say no one, but I feel is not paid enough attention to, and then building an actual development team that can balance the regulatory and clinical, et cetera, et cetera.
All right, great. Thank you.
You know, it was about 50 years ago we went to the moon, right? And I've heard several times during the conference people talking about escape velocity. And I can envision a cartoon, you know, in next year's pamphlet. And there's these kids sitting around the table with their little model rockets and saying, you know, we're going to put it in orbit, escape velocity, you know. Right. So the problem, I think Keith said it exactly right. I mean, when we launched a real effort to go to the moon and put some people in orbit and all that, and in orbit around the moon, it was a really powered effort, people-wise, money-wise. You know, when the space capsule burnt up and all these tragedies, there was plenty of capital to solve the problem and get the thing back on track. And this field, as we were saying, in terms of quality control and drug delivery or cell delivery and all these issues take people and failed efforts and hard work and engineering. And so I think Keith's exactly right, Keith. We've got to find a way to adequately fund this. Otherwise, 50 years from now, we'll still have our little model rockets instead of being escape velocity.
All right, great. Thank you very much. So for the sake of time, we will end our discussion here. I would like to thank the panel members for sharing their knowledge and their time. And we are ready for a break. So thank you very much for your attention. Thank you.